An amoeba is one of nature's most fascinating single-celled organisms. What makes it truly special is its remarkable ability to constantly change its shape. Here we see a typical amoeba cell. Notice its irregular, blob-like shape with a nucleus at the center and various small organelles scattered throughout. An amoeba is defined as a single-celled organism with the unique ability to alter its shape by extending and retracting temporary projections. These temporary projections are called pseudopods, which literally means false feet. Watch as our amoeba extends these pseudopods to change its shape and move around. These pseudopods allow the amoeba to move, capture food, and interact with its environment. The cell can extend them in any direction and retract them as needed. Amoebas are found everywhere in nature. They thrive in freshwater environments like ponds and streams, in moist soil, and even as parasites inside other organisms. The key takeaway is that amoebas are remarkable shape-shifting single cells that use their pseudopods to move, feed, and survive in diverse environments around the world. Amoebas are remarkably adaptable organisms when it comes to choosing their living spaces. Understanding where they thrive helps us appreciate their role in aquatic ecosystems. Amoebas are commonly found in freshwater environments. The three main types of habitats where you'll encounter these single-celled organisms are ponds, streams, and lakes. These tiny organisms can be found throughout these aquatic environments, adapting to the specific conditions each habitat provides. Now let's look at a specific example to understand their habitat preferences better. Amoeba proteus, one of the most well-studied species, has a particular preference for living on decaying vegetation at the bottom of freshwater environments. Here we can see Amoeba proteus in its preferred habitat, nestled among the decaying plant material where it finds abundant food sources. But what makes these environments so attractive to amoebas? The answer lies in their food sources. Amoebas thrive in water that's rich in bacteria and organic substances. These serve as their primary food sources, making nutrient-rich environments ideal for their survival and reproduction. The key takeaway is that amoebas are found in freshwater environments that are rich in bacteria and organic matter, which provide the abundant food sources these organisms need to survive and thrive. Want to see amoebas up close? You can actually culture them in a simple laboratory setup. Today, we'll learn the step-by-step process to grow your own amoeba colony. First, let's gather our materials. You'll need a 100-milliliter container, pond water, decaying leaves or organic ooze, a few grains of wheat, and some mud from the pond bottom. Now let's see the step-by-step procedure for setting up our amoeba culture. Step 1. Add pond water to your container. The pond water contains microscopic cysts that will develop into amoebas. Step 2. Add a small amount of mud or organic ooze from the pond bottom. This provides nutrients and may contain additional amoeba cysts. Step 3. Add some decaying leaves or vegetation. These provide additional organic matter that amoebas feed on. Step 4. Add just a few grains of wheat. The wheat provides nutrients that encourage bacterial growth, which amoebas will feed on. Now we wait. Place your culture in a cool, dark place and check it after a few days. Here's what happens over the first week. Day 1, your setup is complete. By day 3, bacteria begin to multiply. Around day 5, the amoeba cysts start to activate, and by day 7, you should see living amoebas swimming in your culture. Under a microscope, you'll see the amoebas as transparent, blob-like organisms that constantly change shape as they move and feed. The key to successful amoeba culturing is patience and the right environment. The cysts present in pond water will develop into active amoebas when conditions are favorable, giving you a living laboratory to observe these fascinating single-celled organisms. Amoebas have several distinctive characteristics that make them unique among microorganisms. Let's explore what makes these fascinating single-celled creatures so special. First, amoebas are completely transparent. You can see right through them under a microscope, which makes observing their internal structures possible. Amoebas are single-celled eukaryotic organisms. This means they have a true nucleus containing their genetic material, unlike bacteria, which are prokaryotic. The cytoplasm of an amoeba has a jelly-like consistency. It's not liquid like water, but more like a thick gel that can flow and change shape. One of the most remarkable characteristics is their ability to constantly change shape. This shape-changing ability is crucial for their survival and movement. Amoebas have an irregular shape. They don't have a fixed form like other cells. Their cytoplasm is organized into two distinct layers. The contractile vacuole is another important characteristic. This structure helps regulate water content and remove excess water from the cell. Finally, the most recognizable characteristic of amoebas is their ability to form pseudopodia, temporary extensions of their cytoplasm used for movement and feeding. These characteristics, transparency, single-celled eukaryotic nature, jelly-like cytoplasm, shape-changing ability, and pseudopodia formation, make amoebas perfectly adapted for their microscopic lifestyle. Now let's examine the detailed structure of an amoeba cell. These microscopic organisms are incredibly small, measuring only 0.05 to zero point 0.1 millimeters in diameter. The amoeba is surrounded by a semi-permeable cell membrane. This flexible barrier controls what substances can enter and leave the cell, maintaining the cell's internal environment. At the center of the amoeba is the nucleus, which contains the cell's DNA. This is the control center that directs all cellular activities and stores genetic information. The amoeba contains several important organelles. The contractile vacuole helps regulate water balance and removes excess water from the cell. Mitochondria are the powerhouses of the cell, producing energy for all cellular processes. The Golgi apparatus processes and packages materials within the cell. The cytoplasm is divided into two distinct layers. The outer ectoplasm is clear and gel-like, while the inner endoplasm is more granular and contains most of the organelles we just saw. This complex yet elegant structure allows the amoeba to carry out all the functions necessary for life, from movement and feeding to reproduction and waste removal. Pseudopodia are fascinating temporary structures that make amoebas unique among single-celled organisms. These arm-like projections are the key to how amoebas move around and capture their food. Here we see a simplified amoeba cell. The magic happens when the cytoplasm inside pushes against the cell membrane to create pseudopodia. Watch as the cytoplasm flows and pushes the cell membrane outward, forming a pseudopod. This process involves the coordinated movement of the cell's internal material. There are four main types of pseudopodia, each with distinct characteristics and functions. Let's examine each type. First, we have lobopodia. These are bulbous, thick projections that are most common in amoebas like Amoeba proteus. They're perfect for general movement and engulfing large food particles. Filopodia are slender, thread-like projections that extend outward like tiny fingers. These are excellent for exploring the environment and detecting food sources. Reticulopodia form complex branching networks that can merge and separate. These net-like structures are particularly effective for capturing small prey over a wide area. Finally, axopodia are long, thin projections supported by internal microtubules. These rigid structures can extend far from the cell body and are used for both movement and feeding. The key takeaway is that pseudopodia are temporary, versatile structures formed by cytoplasm pushing against the cell membrane. Different types serve different purposes, making amoebas remarkably adaptable to their environment. The plasmalemma is the amoeba's most important boundary, a thin, nearly invisible cell membrane that separates the inside of the cell from the outside environment. The plasmalemma has a unique double-layer structure made of lipid molecules. These two layers create a flexible barrier that can bend and stretch as the amoeba moves. Embedded within these lipid layers are protein molecules that serve as channels and pumps, helping to control what enters and exits the cell. One remarkable feature of the plasmalemma is its self-healing ability. When the membrane is damaged or broken, it can automatically repair itself. The lipid molecules naturally move to close any gaps, restoring the membrane's integrity. This self-repair mechanism is crucial for the amoeba's survival. The plasmalemma facilitates the exchange of water molecules between the inside and outside of the cell. This process is essential for maintaining proper water balance. Water molecules can pass through the membrane in both directions, allowing the amoeba to regulate its internal water content and maintain cellular functions. The plasmalemma serves multiple critical functions for the amoeba. It maintains the cell's structure, regulates what enters and exits, and enables the amoeba's characteristic movement through pseudopodia formation. Without this remarkable membrane, the amoeba could not maintain its shape, move, feed, or survive in its aquatic environment. The plasmalemma truly is the foundation of amoeba life. Cytoplasm is like the jelly filling inside the amoeba cell. Think of it as a thick, gel-like substance that fills up the entire space inside the cell membrane and provides a home for all the cell's important parts. The cytoplasm isn't just one uniform blob, though. It's actually organized into two distinct layers, each with its own special job. Let's explore these layers one by one. The outer layer is called the ectoplasm. This is a clear, stiff layer that sits just beneath the plasma membrane. Think of it like a protective shell that helps maintain the cell's shape and provides structure. The inner region is called the endoplasm. This is where all the action happens. It's a granular, more fluid region that contains all the cell's organelles, like the nucleus, food vacuoles, and contractile vacuole. The granular appearance of the endoplasm comes from all the tiny particles, organelles, and stored materials floating around in this region. It's like a busy workshop where all the cell's important activities take place. So remember, the cytoplasm is divided into two main regions: the clear, stiff ectoplasm on the outside that provides structure, and the granular endoplasm on the inside that houses all the cell's organelles and carries out most cellular activities. Inside the amoeba cell, we find a collection of specialized structures called endoplasmic organelles. These tiny powerhouses and support systems work there to keep the amoeba alive and functioning. The nucleus is the command center of the amoeba. This large spherical organelle contains the cell's DNA, which stores all the genetic instructions needed for the amoeba's survival and reproduction. The contractile vacuole acts like a tiny pump, constantly removing excess water from the amoeba. It also helps with respiration and excretion, maintaining the proper water balance inside the cell. Food vacuoles are like tiny stomachs within the amoeba. They contain digestive enzymes that break down food particles the amoeba has captured, turning them into nutrients the cell can use. Mitochondria are the powerhouses of the amoeba cell. These oval-shaped organelles convert nutrients into ATP, the energy currency that powers all of the amoeba's activities. The amoeba also contains other important organelles. Golgi bodies process and package materials, ribosomes make proteins, and lysosomes clean up cellular waste. Each organelle has a specific job that keeps the amoeba healthy. Together, these endoplasmic organelles work as a coordinated team. The nucleus controls everything. Mitochondria provide energy, vacuoles handle water and food, while other organelles support these vital processes. This cellular teamwork is what makes life possible for the amoeba. Amoebas have a fascinating way of moving that's completely different from how animals with legs or fins move. They use a process called amoeboid movement, which is like crawling at the cellular level. First, the amoeba senses the direction it wants to move. This could be toward food, away from danger, or following chemical signals in its environment. Next, the amoeba forms pseudopodia. These are temporary, blunt-ended projections that look like thick fingers extending from the cell body. The word pseudopodia literally means false feet. Here's where the magic happens. The cytoplasm, the jelly-like substance inside the amoeba, begins to flow from other parts of the cell into the newly formed pseudopod. This is like squeezing toothpaste from the back of the tube. As more and more cytoplasm flows into the pseudopod, it grows larger and extends further. This creates a pulling effect that draws the rest of the amoeba's body forward. It's like the pseudopod is leading and the body is following. This type of movement might seem slow to us, but it's perfectly suited for the amoeba's microscopic world. An amoeba can move about 1 to 5 micrometers per minute. To put that in perspective, that would be like a human crawling just 1 meter per hour. This creeping movement allows amoebas to navigate their watery environment, find food, and escape from threats. It's one of the most primitive forms of locomotion, yet it's incredibly effective for single-celled life. Understanding how pseudopodia form requires looking at the molecular machinery inside the amoeba cell. This process involves several key proteins working together in a coordinated dance. The formation begins with actin microfilaments. These are thin protein fibers that act like the cell's internal skeleton. When the amoeba needs to form a pseudopod, actin filaments start assembling near the cell membrane. These actin filaments work together to push against the plasma membrane from the inside. Think of it like tiny molecular springs pushing outward, creating a bulge in the cell membrane. As the membrane bulges out, cytoplasm begins to flow into this new space. This is called cytoplasmic streaming. The liquid contents of the cell flow like a river into the forming pseudopod. Microtubules act like highways that guide this process. These larger protein tubes help organize the direction of cytoplasmic flow and maintain the cell's internal structure during pseudopod formation. Myosin proteins act like tiny molecular motors. They work with actin to create the contractile force needed to squeeze cytoplasm forward and help coordinate the entire pseudopod formation process. When all these components work together, the result is a fully formed pseudopod. The membrane extends outward, filled with cytoplasm, creating the characteristic arm-like projection that allows the amoeba to move and capture food. This dynamic process allows amoebas to constantly reshape themselves, enabling them to navigate their microscopic world, hunt for food, and escape from threats. It's a beautiful example of how molecular machinery creates complex cellular behavior. Amoebas are fascinating single-celled organisms that need to eat to survive, just like all living things. But how does a microscopic blob of cytoplasm find and consume food? Amoebas exhibit what scientists call holozoic nutrition. This means they ingest solid or liquid food particles, unlike plants that make their own food through photosynthesis. Amoebas are not picky eaters. Their diet includes bacteria, algal cells and filaments, other protozoans, small metazoans like rotifers and nematodes, and even dead organic matter. The nutrition process in amoebas involves 5 key steps. First is ingestion, where the amoeba captures and takes in food. Then digestion breaks down the food particles. Absorption allows nutrients to enter the cytoplasm. Assimilation uses these nutrients for energy and growth. Finally, egestion removes any undigested waste. When an amoeba encounters food, it extends pseudopodia to surround and engulf the particle. This process allows the single-celled organism to capture prey much larger than typical molecules, making it an effective predator in the microscopic world. Understanding amoeba nutrition helps us appreciate how these simple yet sophisticated organisms survive and thrive in their microscopic world through active hunting and efficient food processing. Amoebas obtain their food through a fascinating process called phagocytosis. This is how they literally engulf and capture food particles from their environment. Here we see an amoeba approaching a food particle, such as a bacterium. The amoeba will use its pseudopodia to capture this food through a step-by-step process. First, the amoeba detects the food particle through chemical signals. The cell membrane begins to extend pseudopodia toward the food source. The amoeba extends 2 pseudopodia around the food particle. These temporary projections of cytoplasm reach out like arms to surround the target. The pseudopodia continue to extend and curve around the food particle, forming what scientists call a food cup. This cup-like structure begins to enclose the food. The pseudopodia continue to extend until they completely surround the food particle. Then, the tips of the pseudopodia fuse together, sealing the food inside. Once the pseudopodia fuse, the food particle is now completely inside the amoeba, contained within a membrane-bound structure called a food vacuole. The ingestion process is complete. This remarkable process of phagocytosis allows amoebas to efficiently capture and internalize food particles from their environment. The food is now ready for the next stage of nutrition, digestion. Once food enters the amoeba through ingestion, the real work begins inside the primary food vacuole. This is where digestion takes place, the process of breaking down complex food molecules into simpler, usable nutrients. The digestion process relies on special proteins called digestive enzymes. These molecular machines are released into the food vacuole where they act like tiny scissors, cutting apart the chemical bonds in food molecules. The environment inside the food vacuole is carefully controlled. Initially, the pH is acidic, which helps activate certain digestive enzymes and begins breaking down proteins and other complex molecules. As digestion progresses, the pH gradually shifts from acidic to alkaline. This change activates different types of enzymes that complete the breakdown process, ensuring that proteins, carbohydrates, and fats are all properly digested. The result of this digestive process is a soup of simple molecules: amino acids from proteins, simple sugars from carbohydrates, and fatty acids from fats. These nutrients are now small enough to be absorbed by the amoeba and used for energy and growth. This digestive process is essential for the amoeba's survival. Without proper digestion, the amoeba couldn't extract the nutrients it needs from its food. The two-stage pH change ensures maximum efficiency in breaking down different types of biological molecules. After digestion breaks down food into smaller molecules, the amoeba must absorb these nutrients to use them for energy and growth. This absorption process happens through simple diffusion. Inside the food vacuole, we find the products of digestion: amino acids from proteins, simple sugars from carbohydrates, fatty acids from lipids, plus water and minerals. Simple diffusion allows these nutrients to move from the high concentration inside the food vacuole to the lower concentration in the surrounding protoplasm. No energy is required. The molecules move naturally down their concentration gradient. The absorbed nutrients spread throughout the protoplasm, where they can be used immediately for cellular processes. However, when there are excess nutrients, the amoeba stores them for later use. Excess carbohydrates are converted and stored as glycogen, while excess fats are stored as lipid droplets. This storage system ensures the amoeba has energy reserves during times when food is scarce. This absorption process is crucial for the amoeba's survival. Through simple diffusion, nutrients move efficiently from the digestive vacuole into the protoplasm, providing the building blocks and energy needed for all cellular activities. Now that the amoeba has absorbed nutrients from digested food, it's time for assimilation, the process where these nutrients are actually put to work inside the cell. Here we see our amoeba cell with absorbed nutrients floating in its cytoplasm. These include sugars, fatty acids, and glycerol, the building blocks the cell needs to function and grow. Assimilation involves two main processes. First, the cell uses these nutrients to build new protoplasm, the living material that makes up the cell structure. The second process involves converting nutrients into energy. Sugars, fatty acids, and glycerol are broken down to produce ATP, the universal energy currency of cells. This ATP energy powers all the cellular activities, from movement and pseudopodia formation to maintaining the cell membrane and carrying out metabolic processes. Let's summarize the key points about assimilation in amoeba cells. First, assimilation converts absorbed nutrients into forms the cell can actually use. Second, these nutrients help build new protoplasm for growth and repair. Third, molecules like sugars, fatty acids, and glycerol provide the energy needed for cellular activities. Fourth, ATP is produced as the universal energy currency. Finally, this entire process is essential for the amoeba's survival and proper function. Dissimilation is a crucial metabolic process where amoeba cells break down complex organic molecules into simpler ones, releasing energy that powers all cellular activities. Inside an amoeba cell, dissimilation occurs primarily in the mitochondria and cytoplasm. This process is different from digestion. While digestion breaks down food particles, dissimilation breaks down the cell's own stored molecules. Dissimilation starts with complex molecules like proteins, carbohydrates, and fats that the amoeba has stored from previous feeding. These large molecules contain chemical bonds that store energy. When these complex molecules are broken down, they form simpler molecules and release energy in the form of ATP, the universal energy currency of cells. This is where the amoeba gets the power it needs to function. The ATP energy from dissimilation powers all the amoeba's essential activities. It fuels movement through pseudopodia formation, supports growth and cell maintenance, and provides energy for reproduction when the cell divides. Think of dissimilation as the amoeba's internal power plant, constantly breaking down stored molecules to generate the energy needed for survival. Without this process, the amoeba would have no power to move, grow, or reproduce. Remember, dissimilation is the amoeba's way of converting stored chemical energy into usable power, making it one of the most important metabolic processes for cellular survival. After an amoeba has digested its food, there's always some material left over that can't be broken down or used by the cell. This undigested waste needs to be removed through a process called egestion. The food vacuole containing undigested waste moves toward the cell membrane. The amoeba doesn't have a permanent opening for waste removal like we do. Instead, it creates temporary openings in its flexible cell membrane. When the food vacuole reaches the cell membrane, a temporary opening forms in the ectoplasm. This opening appears right at the spot where the vacuole touches the membrane, creating a direct pathway for waste removal. Now the waste particles are expelled from the cell through this temporary opening. The undigested material is pushed out into the surrounding environment, keeping the amoeba's interior clean and healthy. After the waste is expelled, the temporary opening closes up. The amoeba's flexible membrane seals itself, and the food vacuole, now empty, may be reabsorbed into the cytoplasm or used again for future feeding. This digestion process is crucial for the amoeba's health. Without efficient waste removal, toxic materials could build up inside the cell, interfering with normal cellular functions and potentially harming the organism. Amoebas have a fascinating way of creating new life. Unlike many organisms that need a partner to reproduce, amoebas reproduce asexually, meaning they can create offspring all by themselves. There are three main ways amoebas reproduce asexually. Each method allows them to create new amoebas without needing to find a mate. Let's start with binary fission, the most common method. This is where one amoeba literally splits into two identical daughter cells. Here we see a parent amoeba with its nucleus clearly visible. The process begins when the nucleus starts to divide. First, the nucleus undergoes mitosis, creating two identical nuclei. This ensures each daughter cell will have the same genetic material. Next, the cytoplasm begins to pinch inward, creating a constriction that will eventually separate the two halves. Finally, the constriction completes, and we now have two identical daughter amoebas. Each one is a perfect copy of the original parent. The key takeaway is that binary fission creates two genetically identical amoebas from one parent. This is the most common form of amoeba reproduction. Now let's look at multiple fission, which happens when conditions become harsh or unfavorable for the amoeba. When amoebas face harsh conditions like drought or extreme temperatures, they form a protective cyst around themselves. Inside this cyst, something amazing happens. The nucleus inside the cyst divides multiple times through mitosis, creating many nuclei within the same cell. When conditions improve, the cyst breaks open and multiple daughter amoebas emerge. Instead of just two, we now have many new amoebas from one parent. Multiple fission is nature's way of ensuring survival during tough times. One amoeba can create many offspring, increasing the chances that some will survive harsh conditions. The third method is sporulation, another survival strategy used during unfavorable conditions. In sporulation, the nuclear membrane completely breaks down, releasing chromatin blocks throughout the cytoplasm. Each block will develop into a new nucleus. Each chromatin block develops its own nuclear membrane and cytoplasm forms around it, creating multiple new amoebas. This process can produce many offspring from a single parent. To summarize, amoebas have three remarkable ways to reproduce without a partner: binary fission for normal growth, and multiple fission or sporulation as survival strategies during difficult times. This flexibility helps amoebas thrive in many different environments. Binary fission is the primary way amoebas reproduce. This asexual reproduction method allows a single amoeba to create two identical copies of itself. We start with a single amoeba cell containing one nucleus. The nucleus holds all the genetic material that needs to be copied during reproduction. The first step in binary fission is DNA replication. The nucleus undergoes mitotic division, creating an exact copy of all genetic material. Next, the cytoplasm begins to divide. The cell membrane starts pinching inward at the center, gradually separating the two nuclei into distinct compartments. Finally, the division completes. The single amoeba has now become two identical daughter cells, each containing a complete copy of the original genetic material. Each daughter cell is now a complete, independent amoeba. They are genetically identical to the original parent cell and to each other. This process allows amoebas to rapidly increase their population when conditions are favorable. When amoebas face unfavorable conditions like drought, extreme temperatures, or lack of food, they have a remarkable survival strategy called multiple fission and encystment. Here we see a normal, healthy amoeba with its characteristic pseudopodia extended for movement and feeding. But what happens when the environment becomes hostile? Environmental stresses like extreme temperatures, drought, or toxic substances can threaten the amoeba's survival. When these conditions arise, the amoeba must act quickly. The first step is pseudopodia withdrawal. The amoeba pulls in all of its extended pseudopodia, becoming more compact and spherical to conserve energy and reduce surface area. Next, the amoeba secretes a tough, protective wall around itself, forming what we call a cyst. This cyst acts like a fortress, protecting the cell from harsh external conditions. Inside the safety of the cyst, something remarkable happens. The nucleus begins to divide multiple times through mitosis, creating many copies of the genetic material. Around each nucleus, cytoplasm organizes to form individual daughter cells. This process can produce anywhere from 4 to several dozen new amoebas, depending on how many times the nucleus divides. This strategy provides several survival advantages. Multiple offspring increase the chances that some will survive. The cyst can remain dormant for months or even years, and when favorable conditions return, the population can recover rapidly. When environmental conditions improve, the cyst wall breaks open, releasing the new daughter amoebas to continue the life cycle. This remarkable process ensures the species survives even the harshest conditions. Multiple fission and encystment represent one of nature's most effective survival strategies, allowing these simple organisms to persist through environmental challenges that would otherwise be fatal. Amoebas are single-celled organisms that need to breathe and remove waste just like all living things. However, they accomplish these vital functions in remarkably simple ways. Respiration in amoebas occurs through a process called diffusion. The amoeba has no lungs or gills. Instead, gases pass directly through its plasma membrane. Oxygen molecules from the surrounding water move into the amoeba where oxygen concentration is lower. This happens naturally due to the concentration gradient. At the same time, carbon dioxide produced by cellular respiration moves out of the amoeba into the surrounding water where its concentration is lower. Excretion in amoebas involves two main processes. First, the contractile vacuole removes excess water that enters the cell. The contractile vacuole fills with water and then contracts, pumping the excess water out of the cell. This prevents the amoeba from bursting due to water pressure. Second, metabolic waste products simply diffuse out through the plasma membrane into the surrounding water, similar to how carbon dioxide exits during respiration. The beauty of amoeba physiology lies in its simplicity. These single-celled organisms accomplish essential life functions through basic physical processes, no complex organ systems required. While most amoebas are completely harmless microorganisms that play important roles in their ecosystems, some species can cause serious diseases in humans. The first dangerous species is Entamoeba histolytica. This parasitic amoeba causes amoebic dysentery, a serious intestinal infection that affects millions of people worldwide, particularly in areas with poor sanitation. Amoebic dysentery causes severe symptoms including bloody diarrhea, intense abdominal pain, and dangerous dehydration. The infection spreads through contaminated food and water. Even more dangerous is Naegleria fowleri, commonly known as the brain-eating amoeba. This deadly organism enters the body through the nose, typically when swimming in warm freshwater lakes or rivers. Naegleria fowleri infections are extremely rare, with only a few cases reported each year. However, the infection has a devastating fatality rate of approximately 97%, making it one of the deadliest parasitic infections known. Prevention is key for avoiding these dangerous amoeba infections. Avoid swimming in warm, stagnant freshwater. Always use clean, treated water for drinking, and practice good hygiene. Remember, while these diseases are serious, most amoebas are completely harmless, and these infections remain relatively rare.